All right, good morning, everybody. Obligatory puppy shot. Puppy 1 and puppy 2 should be down there somewhere. They're all flaked out from their walk this morning. All right, so I was having to think about this during lab yesterday, and because lab, a big part of it is going through Mendelian genetics and the chi-squared test. We need to change, or because I'm the professor, I guess, need to change the exam date for next week. So the exam was originally scheduled for 14th. And so I'm going to move it to the 16th. I'll type that up. And I'll send out, I'll upload an updated schedule after class where we kind of, I'll sort out what's going to move around as a result. And so the reason for that is because of, this funky lab schedule that we have. I don't want to have the exam before some of the people have not had that in lab, right? Because that would be unfair to them. And so I want to make sure everybody has an equal treatment, and that will mean moving the exam to Thursday. So that's just kind of a wrinkle that I didn't really foresee when I made the schedule. So we'll do that. And the plus side is A, you get more time to study. Woohoo! Who doesn't love more time to study? And B, we won't have to go like bat out of hell through mitosis and meiosis today, because we'll have time to do that on Tuesday. And so that's a relief to you, and it's a relief to me as well, because I would have to speak like an auctioneer to be able to get through all of that. Okay, so I'll sort that out after class. Okay, so where were we at? Dum, dum, dum. Oh, there you go. There's the schedule. Okay, chat. Get that up. So I'm sure I don't need to tell you this, but I'm going to tell you anyway. You should already be studying pretty heavily for the exam next week, if you aren't already. So hopefully you've been spending time each week going over stuff that we learn, doing questions, practice, stuff like that. But if you haven't done that for whatever reason, you should start doing all of that intensively now, because there's just too much material to study in a couple of days. Right. I know you have lots of other things on your plate, but doing well in the exams is pretty important for getting a good grade in genetics. Okay, so all right, that's where we're at. So I gave you a bunch of definitions last class, like some of the things that we're going to be working with. This is all about the eukaryotic cell cycle. So the prokaryotic one is kind of similar, but it's a little different in the sense that M phase and cell division are not really quite the same, right? They don't have any spindle formation to pull apart their chromosomes, because they just have simple circular chromosomes. Anyway, so the point that really I want to get, or points about the cell cycle, is actually, yeah, there's more than one, two even. Most of the time in the cell cycle that is involved in cell division, right? So this whole G0 over here is like sometimes called the stationary phase. essentially when the cells are not dividing, right? So if we just exclude all the time that cells may not be dividing, which for some of them, like your neurons, that's your entire life, right? As soon as they're born, that's it, they're done. They're never going to divide again. They're called postmitotic. Other cells like your gut or skin, hair, things like that, blood cells, liver, those may divide again, right? But if we exclude all of that non-dividing part of cell cycle out of it, most of the cell cycle is spent both in time and effort in preparing for the M phase and cytokinesis. Right, so if you actually break down, like in a pie chart like this, the actual time spent on the different stages, the vast majority of it is spent on interphase. And interphase is all of this, G1, S phase, and G2 phase. And this is all about getting the cell ready, both in terms of size, stuff like proteins and other things that are needed, and replicating the DNA, right? All of that happens in interphase. M phase is really fairly fast, right? It's just about, lining up the chromosomes and pulling them apart and then dividing the cell. It's pretty straightforward. Most of the time is spent in interphase. And so, sorry, my ears kind of getting blocked up. For like a mouse cell, for example, it's about 24 hours, give or take, for one complete round of the cell cycle, from entering the cell cycle, which will be, around here, to ending up with 2 identical daughter cells. And of that, I think perhaps an hour or 2 are spent in M phase. Okay, so that's one point that's really worth making. It's kind of an important one, right? Because we talk about mitosis, but really the actual mitotic part is a very small part of cell division. Another key point is that there are several checkpoints throughout the cell cycle. These are really, really important. So essentially the cell cycle checkpoints prevent uncontrolled cell division. So those post-mitotic cells that I was telling you about, they are done dividing, right? Because these checkpoints are locked down in those cells. They can never be activated again, right? Which is really, really important, because you don't want neurons suddenly just dividing, right? It would just cause havoc. Also, cells only divide when they're told to. Right? So a cell have to receive a signal, typically an external signal, which says, you divide. The cell goes, all right, and gets busy dividing. Right? So sometimes those signals can be produced in error, right? Either because something's gone wrong in the control of their expression, or even like external factors which mimic, right, those signals, right? So the presence of different growth hormones, for example, in the water supply, right? You don't want those telling different tissues that it's time to get busy again and start dividing. So that's one role of the cell cycle checkpoint. Another one, and there are probably more than just what I'm telling you, these are just the kind of the main ones, is to prevent cell division, or prevent division of a cell that is either not ready or is damaged. So cells have to, if they begin the process of cell division, they have to get all the way through, right? It's an all or nothing deal. You have to go, once you enter it, you have to pop out the other side, otherwise you're told to die, which is something called apoptosis, or programmed cell death. And apoptosis is actually a pretty cool process. And it's one way in which your neurons are pruned. It's like, you know, working on a rose bush. Sorry, I think my allergies are kicking in. Or it's just being surrounded by cats and dogs all the time. Yeah, that might be part of it. So, just like a quick aside, right? Because, you know, it's just cool talking about this stuff. I mean, you're all adults. As adults, you have between a half and 2/3 of the neurons that you started with. So when you're born, you actually have more neurons, brain cells, than you do now, which is kind of crazy, right? And so what happens is the ones that aren't used, that don't make connections with other neurons and don't get stimulated, they kill themselves. It's called pruning or neuronal pruning. And they do so by a process called apoptosis. So it's a really important part of development as well. And so not ready or damaged could mean different things, right? And typically what the checkpoints look for, at least the G1 and G2 checkpoints. So the G1 to S checkpoint, and so this is basically what, pardon me, allows the cell to progress through into the S phase. Because they're pretty much kind of, you know, they're checking that something has been done or that something is ready, right? And so the G1 to S checks for, oops, not FNA, DNA damage, right? Like large-scale mutations, deletions, broken chromosome, broken or missing. And for sufficient nutrients. Right, because you really want to stop the process like early. If it's not going to make it all the way through, you don't want to waste a whole bunch of time and energy getting through to the M phase and then going, oh crap, not going to make it, poof, and then you die, right? So you want to catch this pretty early. And particularly the DNA damage deal is very, very important, right? Because if your DNA is damaged in some way, then if that cell's allowed to go through DNA replication, then you're going to incorporate that damage into the daughter cells. And so every cell that then is descended from that lineage, that original cell, will have that damage incorporated into it. And you can pretty quickly see how that's a bad idea. So once all the machinery has been made, there's enough nutrients, and the DNA is verified as being intact, then the cell can pass into the S phase, which is all about DNA synthesis. So another quick aside, has anyone heard of the BRCA1 and 2 genes? If you're a lady, you're much more likely to have heard of these than if you are a guy. Has anybody heard of BRCA1 and 2? Angelina Jolie is actually a quite commonly used kind of case study, I guess. Anybody know what those are all about? No worries if you don't, but just wanted to see if anybody does. Yeah, Michael, exactly. They're related to breast cancer, right? So if you have if you're homozygous recessive for BRCA1 and 2, I think the probability is around 90%, 80 to 90% that you'll get breast cancer. And Angelina Jolie is often used as an example because you can get genetic screening for that too, particularly if you have a familial history of breast cancer. But she has that history, and she also has those recessive alleles, and so she had a double mastectomy to essentially head off the risk of having breast cancer, because it was almost not certain, right? Because nothing is ever certain, but very highly likely. So BRCA1 and 2 are involved in DNA repair. So if DNA damage is detected at the G1S checkpoint, BRCA1 and 2 are involved in fixing that damage before that cell continues on into DNA replication, DNA synthesis. So if you have mutations in that, essentially that damage isn't fixed, and so there's a risk that that cell slips through that checkpoint and essentially begins uncontrolled cell division, right? Which is largely the definition of cancer. So I thought that's always a kind of a cool kind of real life example to use, because BRCA1 and 2 aren't cell cycle checkpoint proteins. They're involved in DNA repair. But if you are missing those functional copies of them, then your risk of getting cancer goes through the roof, essentially. So once everything's checked out, okay, and you really hope that these checkpoints work, because if they don't, that's also another pathway to cancer, essentially. Then the DNA is replicated. So this is where you get your sister chromatids produced. Right, so these are the identical copies of each chromosome, still stuck together. You divide your DNA, and obviously the more DNA they have, the longer it takes, right? Because these enzymes go at a certain pace. So organisms with larger genomes take longer to divide than organisms with smaller ones. So once that DNA replication is finished, now you get the G2 and G1, and the G stands for gap, right? Because they're always thought to be, at least by microscopy, points of the cell cycle where nothing happened, right? Simply because you couldn't see it. So in G2, now it's, well, essentially G1 is all about preparing for DNA replication, pretty much. like getting all the stuff ready that you need for that. I'll type that up too. G2 is essentially preparing for mitosis and cell division, or cytokinesis. Multitasking because I'm rubbing my puppy's belly with my feet as well. Who says guys can't do more than one thing at once? So the second checkpoint, the G2 to M checkpoint, checks for completion of DNA division. Basically. In other words, are all the chromosomes present and copied? Because again, you don't want to go into M phase if you have an incomplete copy of a chromosome or not all of your DNA is synthesized, because otherwise then you end up with missing chromosomes and DNA in the daughter cells. And these checkpoints, at least the G1 to S and the G2 to M checkpoints, are controlled by actually a very, very elegant system of proteins. Won't get into it much, but it's really, really cool how it is controlled. It's basically through like this kind of cyclic wave or oscillation almost of proteins. And so as they peak, you get through the checkpoint and then they drop again. And then they peak again and you get through the next checkpoint. And I know some about that. I know what proteins are involved. I don't fully understand how they're controlled. Even though that is part of some of my research. It's just quite complex. Okay, and then the final checkpoint is the metaphase checkpoint or the spindle assembly checkpoint. And this is a very interesting one. This one checks to make sure all the chromosomes are aligned on the metaphase plate. Let's see if I can. Sorry, I'm looking for something. I just want to try it out. No, don't want to do that. Hey, that's cool. Can you see what I'm doing? I'm not sure this is like going to be a super productive way of doing anything. But essentially, where can I do it? Let's move this over here. I'm just kind of playing around, but there is a purpose to this. All right, so if that's your cell, there you go, doesn't have a hole in it. And I'm doing this with a trackpad too. I hope you appreciate the mad skills that's going on in front of you right now. Man, that's actually pretty neat. All right, so those are our centrosomes, right? These are the structures from which the spindle fibers are generated, right? And so essentially you have, oh, this is so cool. They're supposed to be straight, but you get the idea. Right, you have all your different chromosomes. This is fun. This is like doing finger painting in class. There you go. All right. This is kind of a shitty picture, but it'll do. And largely it's because I'm too lazy to get all the stuff out to put up my whiteboard because I forgot to do it this morning. So essentially early on in M phase, each chromosome has, or is supposed to have, a spindle fiber attached to either side of it, to the kinetochore, right? And so let's see if I can write. That'll probably be a no. All right, I'm not going to do this again. This is just painful. This is like signing like your name on those things online. Don't know if you've ever done that. Never looks like your signature. Anyway, that's supposed to say centrosome. So essentially the idea is that just by simple mechanical means, these chromosomes are going to get pulled into the center of the cell, right? Because the plane of division is down the middle, equidistant between each centrosome. This is a kind of a sort of like an imaginary line called the metaphase plate. And so essentially you're going to be pinching off the cell at this point. And so if these chromosomes aren't at that center point to get pulled into each side of the cell to become a daughter cell, you end up with the wrong number of chromosomes. So if anaphase, and we'll get to these different phases in a little bit, if anaphase was allowed to proceed now, right, say this checkpoint doesn't work, then this cell would get this chromosome, 2 copies of that one and 1 copy of that one. And this cell will get the opposite, right? Which is pretty catastrophic. You'd be missing entire units of information. Right? And so let's see if I can scrub that out. Oh, that's cool. I'm not sure I want to scrub quite that much out, but never mind. All right, let's go back again. No, draw. There we go. So essentially what the M phase checkpoint is checking for is, I should probably do this in like different colors, but that might be a, oh, how cool is this? I do hope you're all impressed. Put this in your SRIs, okay? Dr. Crook is the bomb at finger painting. Good, right, remember that. I'll be looking for those comments. Okay, so what the M phase checkpoint checks for is, are all the chromosomes aligned on the metaphase plate? And it actually does this in a really kind of cool way. It's looking for tension. It's not looking for their actual physical presence, right? Because how would you know that they're at the center of the cell, right? I mean, the center of the cell doesn't have a sign saying like, you are here. What they're actually, what that checkpoint is a series of proteins on each of those chromosomes that says, yep, I'm being pulled equally from both sides. And if you're being pulled equally from both sides, essentially, if you have the same length piece of string on both sides, you're in the middle. right? It's actually a very elegant solution to the problem. And so the M phase checkpoint checks that that is the case, and it will prevent anaphase from occurring until it gets essentially a tick for each of those chromosomes. Now, not necessarily in mitosis, but in meiosis, can anybody think of a syndrome that occurs when that checkpoint doesn't work properly? Just have a think. This is kind of trying to get you to think about like the application of this stuff, because that's what's cool about this. It's not just the stuff itself, it's how it works. It's actually a fairly common birth defect, about 1 in 1,000 births. And it's something that you can screen for during pregnancy too, fairly early on. Yeah, Maya, good stuff. That's exactly where I was heading. So Down syndrome is the result of this checkpoint failing. And failing in the sense that essentially during mitosis, if I can, I could use different color. Let's use purple. During meiosis, which in females is actually paused, right? So this is actually a pretty funky kind of thing. Essentially, if you're a female, if you're a woman, all of your oocytes, all of your future eggs, right, were made while you were in your mother's uterus, while you're a fetus. And they're all paused at the first stage of meiosis, and they're paused in metaphase, right? And so if that checkpoint breaks down, pardon me, for Down syndrome, it's, is it 21 or 23? I think it's chromosome 21. Chromosome 21 might be off to one side. And so essentially what happens is that egg or sperm, but it's a little more common in females, but it also happens in men as well with increasing age. But that chromosome 21 is stuck over on one side of the cell. So when that turns into an egg and is fertilized, that will have 2 copies of chromosome 21, not 1. And so you get the copy from the sperm from the father, and that will give you the 3 copies of chromosome 21, or trisomy, and that leads to Down syndrome. So this is a big deal, right? That this checkpoint works, because if it doesn't, you get incorrect numbers of chromosomes, which is called aneuploidy. So there you go. That's kind of one way to put this stuff into context, right, which always makes it more interesting to learn. Makes it easier to learn too. All right, now let's see if I can avoid just drawing and dribbling all over the rest of my PowerPoints. Isn't that cool? I'm so impressed myself. I'm going to do that like all the time now. I see if I can draw a cat. Anyway, cool. So that's the checkpoints, right? So this is, and one of the reasons for spending so much time going through this is because I really want you to get the sense that this is a very tightly controlled process, right? With multiple steps. It's not just the division of DNA in M phase. There's an awful lot that goes into this, and it is truly a fascinating process. It really is. And it's something that I just love thinking about and teaching about because it's just super, super cool. All right, we're only on slide 4. Better get my finger out, so to speak. All right. Bugger. Okay, I gotta get rid of this because otherwise that's just going to annoy me. There you go. That would be the downside of. Oh, I don't have to do that. This is, oh, okay. Maybe I'm not going to do this in future because now I've got to delete every single bloody line. Or I'm going to have that smiley face just sticking there. All right. Sorry, everybody. Not going to do this again. Fun as it was, like this is going to take me all class just to, fucking hell. Pardon my French. You would have thought that, oh, hang on. There you go. Classic case of the problem lies between chair and keyboard, right? Anyway. So give you a little bit more stuff on kind of like what the chromosomes look like. So again, this is sister chromatids. The kinetochore is a collection of proteins attached to the centromere, and the centromere isn't like a, I mean, it has some kind of structure, I guess, but it's just part of the chromosome where the kinetochore assembles. Doesn't always have to be in the middle, right? So depending on the organism, you know, the centromere could be up here or down there or bang in the middle. It varies. You've typically got a short and a long arm of the chromosome, and this is actually part of how you refer to where a gene is, right? You refer to which arm it's on. And so the P stands for petite, which is cute, French for small. Q, I don't know what it stands for, probably nothing. And essentially this condensed duplicated form is what you see only in prophase of cell division, because you have, this is like a stylistic version. It doesn't actually look like this in the normal cell, right? A non-dividing cell at least. But after the chromosome has been copied, you have these two sister chromatids attached together, and it's going to be condensed. The reason why you condense your chromosomes is it just makes it easier to pull them apart, right? It's like, my favorite analogy is like serving spaghetti versus serving meatballs. It's a lot easier to count out meatballs than it is strings of spaghetti. Essentially, these two sister chromatids, they are going to be pulled apart during anaphase. And so just to reiterate again, cohesin, not cohesion, is the protein that holds the two sister chromatids together. And it plays an even more important role in meiosis. Stop talking. All right. Any questions so far? I actually forgot to ask if anybody had any questions from all the other stuff that we learned prior to this. Oh. It's certainly going to be sad to go back to teaching in person in some senses, not have my pups around. Okay, so mitosis, division or partitioning of DNA. We want to make sure each of our daughter cells has the same genetic complement or same complement of chromosomes. Oh, really? Sorry, extra puppy shot. There she is. Actually, can you even, yeah. She loves sleeping upside down for some reason. I don't know why. Maybe it's just what puppies do. Okay, so again, remember that's the end result that you want. Mitosis generates two identical daughter cells, two diploid. I'll just type that out because it's worth reiterating, right? This is another point where students get a little bit unstuck, right? Particularly between mitosis and meiosis. Goddamn dog. Sorry, just hanging up in my power cord. And that's a very important distinction to make because meiosis does not. Okay, so essentially there are a bunch of stages or phases in mitosis, the actual division or partition of DNA. And these, some of them are pretty well defined because of what happens in them. Others are really kind of just a way in which we separate different things because we like categorizing stuff, right? And so prophase and prometaphase, and even metaphase in part, are really part of a continuum, right? The aim of the game in mitosis is to get those chromosomes lined up at the center of the cell and then pull the chromatids apart. That's it, right? That's the role of mitosis. And so prophase, prometaphase, and metaphase are really all the preparation up until that point. Anaphase is pulling them apart, and telophase is just tidying up the mess, essentially. Okay, so if you remember back to, I'm gonna make this a little bigger here, right? So we've gone through G1, preparing for DNA replication. We've synthesized our DNA, right? So we've replicated our DNA in the S phase, and we've prepared all of the stuff, centrosomes, proteins, microtubulin, right, for the spindle fibers in G2. And so this is pretty much what the cell would look like. It's going to be a little bit bigger than a non-dividing cell. It's going to have two centrosomes, right? So these are where the spindle fibers come from. And the spindle fibers are microtubulin. And those are also known as microtubule, if I can spell that right, organizing centers, called mTOCs. So you need two of them because you're going to have one on either side of the cell. That's how you pull the DNA apart, the chromosomes apart, to get equal numbers of chromosomes in each daughter, right? But in the G2, they've simply been replicated, right? So we now have two of these. We still have our nuclear envelope. And even though the DNA is replicated, you can't really tell by looking at it, because it's still in this uncondensed form that you will see in a normal non-dividing cell. So you, again, just to kind of, this is a bit of a misconception about chromosomes, you only ever see those classic X shapes. Yeah, these ones, well, these are actually non-duplicated. So, but anyway, you only see those kind of condensed, like short forms of the chromosomes during M phase. Right? You don't see them at any other point. It's actually, let me see. I want to see if I can find a cool video. Let's see if I can remember where that was from. You have to bear with me for a moment. Ah, come on. No, not central dogma dingbat. That's not what I want. That's something completely different. Ah. It's a long time since I've actually used. Yeah, maybe this is the one. Ah, here we go. Nope, this one. Sorry, it's just a video that has a really cool image of cell division. Bollocks. It's not this one. Oh, pardon my French. Go away. Anyway, never mind. I'll find it and I'll post the link for next class. It's just really neat. Anyway, sorry, getting distracted. Got to focus. It's all that extra free time that I now have. I can just go chanting on about things that I wouldn't have been able to before. Anyway, I'll find that video and I'll show it to you next class because it's super cool. So essentially, to be able to divide the DNA, we got to do a bunch of stuff. What things, there you go, ask the class time. What things do you think that we need to do in able to divide our DNA correctly? Looking at this picture, what things would you have to change from this picture to be able to get to the point where those chromosomes are neatly aligned on the metaphase plate?
Get rid of the nuclear envelope.
Yeah, exactly. That's a really, really good point. So first, you have to get rid of the nuclear membrane. Well, not first, but one of the things you have to do. Right, because how are you going to be able to attach spindle fibers to your chromosomes if there's like something in the way, right? So you got to get rid of that. What else? What else have you got to do? Think about the process. Kind of mentioned one of them a little bit earlier when I was talking about the centrosomes. What needs to form from the centrosomes? Oh yeah, cool. Thank you, Lindsay. Exactly. Right. So 2, the centrosomes have, plural, have to move to opposite, sides of the cell. Yeah, good job. Right, because those are what define essentially what are going to be the daughter cells. One daughter cell will have one centrosome, the other daughter cell will have the other centrosome. What else? Okay, so we've got the centrosomes. How do the centrosomes pull the sister chromatids apart?
Spindle fibers.
Right. Yeah, spindle fibers. Thank you, Mika and Jesus. So spindle fibers have to form, or really assemble, because they're pretty cool structures. I wonder if I can find a picture of. Get out of the way. Actually, the lab, oh, go away. Lab that I did a postdoc next to, they worked a lot on microtubule dynamics, which I think is pretty cool. So microtubules, the spindle fibers, are assemblies of two different proteins, alpha tubulin and beta tubulin. And they form, it's kind of like a churro, basically, one way of thinking about it, right? If you could make churros out of two separate proteins. Yeah, leave your ears on it. Right. And so they form in this kind of spiral form, and they actually have polarity as well, right? They have a plus end and a minus end. Plus end is where things get added to. And here we go. That's a cool picture. So these polymerize, right? So they're actually, the centrosome would be down here at the bottom, and they form kind of out from that. They assemble out from it. It's like a, everybody seen Big Hero 6? Yeah, it's like those little nanobots assembling. That's essentially how it works, right? So that film took some inspiration, I think, at least, from you know, biology. Let's just get that up, right? And so that's also how they shorten, right? So spindle fibers pull physically, like tug of war, on those chromosomes by depolymerizing, right? So they kind of extend out, and then to exert mechanical tension, they actually kind of take bits of that protein out, and that shortens them, and that's what actually pulls on the chromosomes, which is, you know, it's a pretty funky thing to think about. So these are super neat. And you're actually going off on a little bit of a tangent here, just because biology is so cool. These are also the cellular highways. So it depends on kind of what they're, oops, didn't mean to click on that. Get back. These are the, like the ropes or the highways along which stuff is carried in your cells. So if you think about your neurons, right, you would have, let's say, I'll just use my arms because I can't point to my feet. So there's going to be a nerve cell body in my spine, right? One of the ganglia there, that's going to go all the way down my arm to my fingertip. So if I, you know, hit that with a hammer, I'll go, ow, because that's going to transduce the signal all the way back up. It's going to tell my brain that my finger hurts, right? But the way it does that is by proteins that are down here that are produced here, right? In that cell body in my spine. And the way that those proteins are transported all the way down that axon, right? All the way down that nerve process to the end of that nerve, to the synaptic junction, wherever that might be, is using these molecular motors called kinesin and dynein. And they actually walk, they walk like that. They're really cool. Anyway, that's a bit of a tangent, not super relevant to what we're talking about, but fun to talk about nonetheless. All right. Sorry, I'm just having too much fun. Curse it. Who said teaching is boring? Teaching is awesome. Okay, so spindle fibers have to form, centrosomes have to get to the opposite sides of the cells, nuclear membrane has to disappear. There's a fourth one. It's actually in that PowerPoint, in one of the bullet points. What have the chromosomes not yet done? They're still all like these long fibers of chromatin. What do we need to do before we can accurately separate those?
They need to condense.
Yeah, you need to kind of condense them down. That's it, say it exactly. You got to condense them down so that they're easier to partition, right? So you don't have any tangles or errors, right? So essentially, all of what we've just talked about is prophase and prometaphase, right? That's generally all that's going on in those steps. Oh, and also, so let's just and just the last one as well, forgot this one. Spindle fibers have to attach to, pardon me, each sister chromatid. Just forgot that one, kind of important. And so this happens in just a progression, right? There aren't really kind of defined steps in this. It's just a continuum from G2 to metaphase, right? And so prophase is where those centrosomes start pushing themselves to either side of the cell, and the chromosomes start condensing. So who was it that is it Lindsay? Yeah. So Lindsay pointed this out earlier. It's kind of why I'm picking you out. The cool thing is these centrosomes get to opposite sides of the cell by pushing themselves apart. And so they actually use spindle fibers, right? And they kind of basically generate a spindle fiber that pushes the other centrosome away. And so that's how they end up on opposite sides of the cell. It's because they've pushed each other as far away from each other as they can get. And so that's how you get one centrosome on either side. And that's also how you get the plane of cell division or cytokinesis, or even the metaphase plate. So where those centrosomes end up, and this is actually another kind of cool point. Let's see if I can draw this up. So if you have centrosome here and here, you'll end up with a centrosome here and here, because they will have pushed themselves apart like so. I mean, I know I'm not a good artist, but this looks absolutely awful, but hopefully we'll get the point across. However, then you'll have, actually, let's get rid of that. This is a cool point to make because, you know, at least in development, cell division occurs as a continuous process, right? It's not just a one-off. And so you end up with your two daughter cells, right? Each one has centrosome either side. And let's take that one on the right. Now, when it divides again, each one of those centrosomes kind of pushes each other apart, right, to this kind of opposite sides of the cell. And so then you'll get, kind of using up a lot of space here. The cell division plane will change. So originally, you know, the centrosomes are at the top, like they are here. They'll push each other to the sides, and then you'll get the cell division occur like so, right? Now that centrosome is on this side, like so. Now you'll get centrosome at top and bottom, and now the plane of cell division rotates. And so when you see those early animations of cell division in an embryo, and you'll always hear them go, oh, boop, and then each one of those go boop, like that. Calm down. That's why. That's how you get that ball of cells in an early embryo having that shape. It's not just like one long string of cells, because every time you go through cell division, the plane of cell division rotates by 90 degrees. It's just another cool thing to talk about. I'm having so much fun with this annotate thing. Anyway. So that continues from prophase, just getting this into the right place. And part of the merit for kind of going through mitosis in some degree of detail is because the same principles, by and large, are involved in meiosis. It's just meiosis is just far more complicated. Prometaphase, the nuclear membrane disappears. You can just kind of see it, you know, over here. The chromosomes condense even further, and these spindle fibers are now randomly attaching to the kinetochores. Now, this is a random process. It's literally like kind of getting a harpoon gun, like fire it off into the darkness. You know, maybe you'll hit a chromosome, maybe you won't. If you hit a chromosome and it attaches, that spindle fiber is stabilized. If it just misses, it depolymerizes and you start again. Right, so this takes a while because you need to have a spindle fiber on both kinetochores of a chromosome, both sister chromatids. If you don't, like this one up here, that spindle fiber will then pull that chromosome all the way over here, and you'll end up with an aneuploid daughter cell. Right. So this takes a while, and it is random. It is not a guided process. These spindle fibers don't know where they're going. They're just polymerized out. And if they hit something, yay. If they don't, they just disappear and start again. Right. It's a totally random process. And so this takes a while. And this is one of the reasons why that mitosis or M phase checkpoint exists, because this is a random process. You also sometimes have two spindle fibers on one side and one on another, so that's also going to result in kind of a misalignment of the chromosomes. And so that essentially jockeys around, right? And it is a dynamic process. You can actually see like these chromosomes jiggling around until they all end up aligned on the metaphase plate. And this metaphase plate, at least at this point, doesn't have anything that says this is where it is. The only reason why it is there is it's because it's equal distance between one centrosome and the other. That's it. It's halfway in between. Now the M phase checkpoint, what it's looking for is equal tension on both sides of the chromosome. Because if you have equal tension, that means the chromosome's in the middle of the cell on the metaphase plate. Does that make sense? It's kind of a really important point to get. Just because until that requirement is satisfied, meiosis is the same as well, cannot proceed. It will just wait there until, you know, all those chromosomes have had spindle fibers attached and jiggled round until they end up on the metaphase plate. Now, once that requirement has been satisfied, the anaphase promoting complex, because it's a whole bunch of proteins, activates separase. Remember I talked about that last Tuesday? And that cleaves cohesin. Sorry, typo. Cohesin is the thing that sticks those sister chromatids together. And so basically it's like you've got a tug of war, two people that are glued together, right? Ropes attached to each person. Dissolve that glue and you're constantly pulling on those, right? So it's a dynamic process. It's not that they're just sitting there. They're there because they're being pulled equally by both sides or both centrosomes. So as soon as that glue goes, as soon as that cohesin is destroyed, they get pulled apart, right? And that's anaphase. Anaphase is super fast. It's really simple. All that happens is the APC is activated, separase cleaves cohesin, and the existing tension created by those spindle fibers pulls the chromatids apart to either end of the cell or pole. And basically, telophase is cleaning up the mess. So all those things which we saw happening up here, the condensing chromosomes, the spindle formation, the dissolution of the nuclear membrane, all of that has to be reversed, because otherwise you'll have a daughter cell which doesn't look like a daughter cell, right? It looks like a cell that's still going through M phase. So during telophase, and telophase and cytokinesis more or less happen at the same time, right? During telophase, you reverse all of what happened in the early stages of M phase, right? These centrosomes relax, not centrosomes, chromosomes relax, they decondense. The nuclear membrane reforms, the nucleolus forms. The spindle fibers depolymerize and disappear, right? And typically while that's occurring, the cell starts getting pinched off. It's called a cleavage furrow. And this actually occurs, it's literally like taking a balloon and you just kind of squeeze it in the center until you have like a balloon and a balloon, and then just go boink, like that. And now you have two balloons. That's essentially cytokinesis. A little more complicated than that, I guess. And it happens just here because of the proteins that are sort of put in place while the metaphase plate exists, right? So if we go back to metaphase, pardon me. So, you know, I said that there's nothing that actually says, here is the metaphase plate. But once those chromosomes start assembling, you know, and kind of moving around and kind of forming on this imaginary plate, it actually results in proteins being put down or locating there. I think one of them is called Aurora, but I might be wrong. And those essentially say this is where we're going to form the cleavage furrow. This is the cytokinesis division point. And so that essentially thence, and it pinches off the membrane, and so you end up with two daughter cells, like so. And that's what it actually looks like, which is pretty cool. Obviously, this is in an animal cell. In plant cells, it's a bit different because they have rigid cell walls. So in plant cells, they actually have, well, the metaphase plate serves as a location for the new cell wall components to be laid down, right? So you see these vesicles, essentially those are little mini bits of cell wall, essentially. And that forms the cell membrane there, because plants have plasma membranes too. And that also serves as the location of the cell wall to be synthesized. What's in there? Yeah, that's pretty much the same deal. Pretty much just go through the points. I forget. So that's pretty much mitosis. So mitosis is a relatively straightforward process. Obviously, there's a lot going on. We've got all the different phases, G1, S phase, G2, and M phase, cytokinesis. We've got the different checkpoints. You need to know what happens at each phase and what those checkpoints do, like their purpose. And then there's the actual kind of mechanism of DNA partitioning or division, which is M phase. So keep in mind at all points, the outcome is two identical diploid daughter cells. So this is all about somatic or asexual growth. So you're either replacing cells, such as your gut cells or skin cells or whatever that are dying or run out of puff, right? You're growing. I try not to, but you know, I'm getting older, so I do. But this is what results in a baby, essentially, a functioning standalone organism from one single fertilized zygote, right? So mitosis is how we get from there to there as well, with a lot of stuff in between. And it's also how organisms such as yeast and, I mean, bacteria, obviously, and a few other animals, there aren't too many, like some insects reproduce asexually, like Daphnia, water fleas, for example. And so this is not sexual reproduction, but it may be used in asexual reproduction. Meiosis is much more complex. There's a lot more going on. 2 stages, 4 daughter cells, haploid, non-identical, right? So make sure you get this, because we're not going to be going over as much of the mechanism in meiosis because we've covered a lot of it in mitosis. We're really going to be, well, I'm going to be focusing on the stuff that makes meiosis what it is and how you end up with such diversity in sexual reproduction, right? That's really the purpose of meiosis, or one of them. So have a look through that PowerPoint before Tuesday, because it's a doozy. There's a lot going on and there's a lot will just turn your head around. So make sure you understand this and at least have a look through the meiosis PowerPoint ahead of time. All right, everybody, that was a blast, as always. Stay safe, have fun, study hard, and I'll see you on Tuesday.